Hello all, I am Ruchi Gakhar. I am a PhD student at Material Science Department. I am working on biophotovoltaic cells and today I will talk about my research project. First of all, what are photovoltaic cells? Photovoltaic cells or solar cells is device which converts solar energy into electrical energy without use of any external power source. So now the question arises, why do we need photovoltaic cells? We need these devices because we are using fossil fuels and other carbon compounds for many years for our energy generation, which are contaminating our environment and producing effects such as global house warming and ozone layer depletion. Some of my friends here know a lot about fossil fuels and their effects. So we really need to find an alternative source of energy generation. And the best way is to harness some energy from natural resources. Natural resources are like wind and solar power. Among these, solar radiation is the most abundant source of energy. The solar energy we receive in 1 year is about this much. This is about 3.8 million exajoules energy. This is 10,000 times more than the energy we use in 1 year. So what we really need to make a device so that we can convert this energy and use it for our electrical needs. And here comes our solar cells. So how does a solar cell works? It consists of two semiconducting layers. One is rich in electrons and other is deficient in electrons. This is known as anode and this is known as cathode. Between these two layers we have a chemical species we call as electrolyte. It can undergo both oxidation and reduction, so it helps in completing the electrical circuit. So, now when sunlight falls upon the anode, it absorbs some energy and some electrons from the lower shell goes to the higher energy level and they eventually get free. These free electrons enter this external circuit and travel towards cathode. And the electrical circuit is completed with the help of electrolyte and the power is generated across this load. So this is how a general photovoltaic cell works. In order to increase its efficiency, people modify this anode in different ways. Some form thin film, some attach some groups onto it, and if biological groups are attached to this anode, then it is known as biophotovoltaic cell. And this is what I am working on. So the idea here is to generate electricity with the use of biomolecules. This idea is not new. Nature has been doing this since many years. We all know about photosynthesis. Plants take their energy from sunlight and synthesize their food. During this process, some energy is also released. And similar to plants, there are some bacteria also available in nature which do the same conversion with the help of some enzymes. So if we take some bacteria and we can attach onto this anode, then we can form a device and most of our environmental problems will be solved by the nature itself. But this is not very easy, because to keep the bacteria alive into a device is really difficult. Some of the research groups have isolated the enzyme inside the bacteria and they have attached this enzyme onto this anode and they have formed a cell. But the problem is that such kind of devices are not very stable, because the enzyme becomes inactive after some time as it is not getting nutrition from the whole cell. So it is really important that we use the whole bacteria as a whole. And this is what I am working on. I am planning to use a bacteria and I am planning to form a device out of it which would be stable and efficient, and I hope that I can solve the environmental problems with this. So that's all. A question? Yeah.
Is there degradation of the bioform? Does the bacteria die after a while?
Yeah, there are many reasons, because bacteria need certain conditions. So the pH should be same, and like my bacteria, it grows in anaerobic conditions, so there should be no oxygen. So it is pretty difficult that we provide sunlight, but there should be no oxygen. So these are the conditions which make this thing a bit of challenge. So if we can do all that thing, then it can be a very efficient device.
How long do the enzymes last in optimal conditions if they were using those?
The research so far is done, the efficiency is less than 1%, and the stability is like what they say is few days or maximum a month. So, and that's not what we want. So.
I mean, if there is no sunlight, how long this bacteria, the biosystem will be stable?
Without sunlight, they can sustain its sunlight. We are giving for them to operate. They don't need sunlight. Means this bacteria don't need sunlight to grow. It needs anaerobic conditions, some pH conditions, and some particular media to grow. So it doesn't need sunlight to grow. But we want it to be stable with sunlight and doing its work.
How often are you going to have to refresh the media in your cell to keep the bacteria colony alive?
We will insert the bacteria into it once they are grown.
But once they're grown, that media that they're grown, they're going to exhaust it. So how often, how viable is this to maintain?
Right now, I don't know about that step because I'm just starting. So right now I am changing the media after 3-4 days. So I don't know how efficiently they will work in the device. So.
I'm just wondering if that energy amount from sun is the average one, or is it averaged? Because it may change during the year, right?
It
Because
may
of the
change.
angle of the sun rays.
I found this in a report in 2010. So I am not sure it is average or it is total or in what form they have.
It's probably a total. That's just how much sunlight strikes the earth.
Is that the upper atmosphere or is that the surface?
It's at Earth's surface. Yeah.
Because clouds or dirt in the air.
Yeah, you are right. It depends upon climate and all the conditions. So I'm not sure. And this is the report from 2010. And I'm not sure by what measure they measure that, but this is what they provided.
I didn't quite catch, where do the bacteria fit into the circuit over there? Is it as the
Actually,
node
what
or the packet?
actually what we are planning to do is we will cross-link our bacteria into a polymer first, and then we will attach it to a semiconducting layer, because we need a semiconductor or metal to complete the circuit, or maybe we can use conducting polymers. So because polymer itself is insulating, that's why we need this thing.
But the bacteria is being used as the electron.
So bacteria will be attached something like that, consider it like this. So the light will fall into these things first, and then they will transfer their, they will do some conversion, and then they will transfer the electron to this anode, and then they will go into circuit. That's how it will work.
How are you getting the electron transferred from the bacteria to the anode?
It's their internal mechanism. There is some enzyme inside this bacteria. Most probably it is hydrogenase enzyme. So.
Right, but inside the bacteria, that electron transfer goes from one molecule to the next. And so how are you going to actually uncouple that process from the bacteria so that electron leaves the cell membrane and, flows through your circuit.
That's why we will cross-link into such a polymer, and then we will find a particular anode, and we will do some studies before that it is doing that electron transport mechanism within. Then we will form the whole device like that.
Sorry.
Yeah.
Well, we don't have time for another person, so we can grill her for another time.
Damn. Yes, I'm going back to the basics one, because if you still use the anode, then what's the purpose to use the bacteria over there?
Yeah, actually, in general also we have to modify this anode, because generally, generally people use TiO2 or ZNO semiconductors here. But the problem is that they are a wide band gap semiconductors. Like TiO2 has a band gap of 3.2 electron volts. So it will absorb only UV range. The wavelength would be less, because energy is inversely proportional to wavelength. If this band gap is high, so wavelength they are going to absorb is very less. So in order to make them efficient, it means that they should absorb the whole spectrum of wavelength. Means they should be able to absorb IR region, UV, and visible whole. So that's why we modify it. Sometimes we dope it with some groups. Sometimes we form a film on that, so that one part of spectrum TiO2 will absorb, and one part of spectrum the attached group will absorb. So that's how this thing works. And sometimes we attach some quantum dots and other species. So we are using biomolecules to increase its efficiency, but we still need some anode here. Bacteria alone can't do that whole process.
Okay, thanks.